Look at the parts of a neuron. A typical neuron has a cell body, nucleus, dendrites, axon, and axon terminals. Let's pause to take a look at what each part does. The cell body and nucleus are not unique to nerve cells, so we'll focus on the other parts. The dendrites are short extensions that branch off the cell body of the neuron. They receive signals from other neurons at the synapses, which are gaps between neurons. The signal is then passed along the axon. These are covered with myelin sheaths, which help the signal move faster. The myelin sheaths are interspaced with the nodes of Ranvier, which the signal skips over. After the signal reaches the axon terminal, neurotransmitters are released into the synaptic cleft between one neuron and another in a process that depends on calcium ion movement.
When the signal reaches the axon terminal, calcium ion channels open. The resulting influx of ions causes the release of neurotransmitters which travel across the synaptic cleft and bind to receptors to either continue or end the signal. Let's examine the cell membrane in more detail. The three main proteins involved are sodium channels, potassium channels, and sodium-potassium pumps. Sodium-potassium pumps help maintain the membrane potential by using ATP to transport ions against their concentration gradient. 3 sodium ions are pumped out of the cell for every 2 potassium ions that are pumped into the cell. Therefore, the inside of the cell is negative relative to the outside, and there is a higher concentration of sodium outside and potassium inside. The sodium and potassium channels function in facilitated diffusion of ions down their concentration gradient, so sodium ions will move in and potassium ions will move out. These channels are voltage gated, which means that they open in response to a change in membrane potential. The membrane is much more permeable to potassium because there are many more potassium channels. This becomes important later.
Initially, the cell membrane is in its resting state. Gated sodium and potassium ion channels are closed, and ungated channels maintain the resting potential. During depolarization, some sodium channels open after they receive a stimulus, resulting in the inward flow of sodium ions. If the membrane potential reaches negative 55 millivolts, the threshold is reached and starts an action potential. More sodium channels open, allowing sodium ions to flow into the cell. This makes the potential less negative until it peaks. The action potential falls as most sodium channels close. On the other hand, potassium channels open, releasing potassium ions out of the cell. There's a slight dip below the usual resting potential as the sodium channels remain closed, but some potassium channels remain open. This is resolved when the sodium and potassium channels become unblocked.
The opening of sodium channels causes the influx of ions, which then causes more sodium channels to open as a result. In this way, the beginning of the action potential proceeds further. Almost immediately afterwards, the resulting hyperpolarization as a result of sodium channels closing and potassium channels opening causes a refractory period, ending the action potential. This refractory period prevents the action potential from traveling backwards. After the action potential moves, the membrane potential resets via the pump. Botulinum is the most acutely lethal toxin known to man and works by blocking the release of acetylcholine, an important neurotransmitter from the synaptic vesicle. First, botulinum binds to the synaptic terminal and is taken up into the neuron via vesicles. Once inside, the toxin enters the cytoplasm and cleaves proteins which facilitate the fusion of synaptic vesicles with the presynaptic membrane. Therefore, the neurotransmitters are not released and signaling stops, causing paralysis.